What's going on, everybody? Ken here, and welcome to the Metropolis. On today's video, we will be following up on the biological focused themes of the last video. This one not having to do so much with CRISPR technology and RNA, this one having to do with some recent advancements in our understanding and manipulation of DNA. Hitherto, we had only been able to store data on DNA, but we hadn't been able to manipulate it. The human body itself with DNA is something that nature has created to store massive amounts of data. The encoding of our genes, all of our predispositions, all of the schematics that make up you, I, and most other biological beings are stored in this crazy system known as DNA. We have been looking to figure out how to store whatever data we want on it and how to manipulate that. Kind of similar to the way that in Metalocalypse they stored their music on water. If you don't get that reference, no worries. If you do, awesome. But this sort of thing is absolutely fascinating to me, and anything that is in the realm of sounding like science fiction, I am a big fan of. We've known about DNA for a while. We've done rudimentary cloning of animals and whatnot, but it has only been recently that we've been able to do this kind of honestly science fiction sounding stuff to DNA. So let's have a look at this article. This article from SciTech Daily will be in the description below, as are all articles that I cover in these videos, if you'd like to have a look at yourself. Is titled, "Scientists Have Found a Way to Manipulate Digital Data Stored in DNA." Now, scientists from the CNRS, which I discovered is the French National Center for Scientific Research, and the University of Tokyo have developed the application of a novel enzyme-based technique, providing the first hints as to how DNA technological challenges may be solved. Nonlinear decision-making with enzymatic neural networks. DNA can be utilized to reliably store massive amounts of digital data. However, it has hitherto proven challenging to retrieve or manipulate specific data embedded in these molecules. Now scientists from the CNRS and the University of Tokyo have developed the use of a novel enzyme-based technique, providing the initial clues as to how these technical obstacles may be overcome. The research was recently published in the Journal of Nature. Nature has unquestionably developed the best method for massive data storage, not the magazine, actual Nature itself. DNA. DNA is the best method for massive data storage, as I mentioned. Based on this knowledge, DNA has been used to store digital data by translating binary zeros and ones values into the 4 DNA letters A, T, C, or G, which is just kind of amazing in and of itself. But how can one search through the database of data encoded in DNA to discover a certain datum? And how is it possible to execute computations using DNA-encoded data without first transforming it into electronic forms? These are the questions that the research teams from CNRS and University of Tokyo and CNRS ESPCI laboratories have attempted to answer. They are experimenting with a new approach using enzymes and artificial neurons and neural networks for direct operations on DNA data. Specifically, the researchers have made use of the reactions of 3 enzymes to design chemical neurons, artificial neurons, quote unquote, that reproduce the network architecture and ability for complex calculations exhibited by true neurons. So basically, as I've understood it, they've somehow chained these 3 enzymes, which in general, just the functional existence of enzymes blows my mind. Not that I'm confused. Obviously, you need something of that nature to come up with this, whatever this is, this reality we have. But the fact that they're that enzymes are, you could consider them widgets perhaps, or just little, if you program, I think they're akin to functions and methods in programming. A little bit of on-call, hey, this thing that has this feature, this thing that has this feature. And it looks like they were able to chain 3 of these enzymes to create artificial neurons and then, I guess, trick DNA into operating or reacting in a certain way that was to their desired effect. Their chemical neurons can execute calculations with data on DNA strands and express the results as fluorescent signals. So that's insane to me. So basically, they have the three enzymes that they stack together, creating these artificial neurons that reproduce the network architecture and ability for complex calculations exhibited by true neurons, tricking the DNA, or at least giving the DNA what it needs to be manipulated in the same way that actual neurons would be. So bypassing the need for any sort of electronic, this is a biological miniature computer. It's a biological hard drive storage device, essentially. I guess it's just read and write, so it doesn't have everything that a computer would have, but it's serving, as far as I can tell, the same functional needs, the same functional applications of a rudimentary computer or a hard drive. Essentially, it is reading and writing, but doing it all biologically, which is insane. So they say how they manipulate it with the three enzymes, and the chemical neurons can execute calculations with data on the DNA strands, express the results as fluorescent signals. So basically, that is, I would imagine, akin to their viewport, their window. They can see what they're working on, or they can, get some sort of they can get some editing ability, retrieval ability, modification or query ability to find specific datums, as they mentioned, which are represented through fluorescent signals, which again, the biological computer nature of this is mind-blowing to me. The LIMMS and the Gulliver teams have also innovated by assembling two layers of the artificial neurons in order to refine calculations. Precision is further enhanced through microfluidic miniaturizations of reactions, allowing tens of thousands to take place. The fruit of a decade of cooperation between French biochemists and Japanese microfluidics engineers, these breakthroughs may eventually permit better screening for certain diseases, as well as manipulation of gigantic DNA-encoded databases. DNA, when kept away from water, air, and light, can be preserved for hundreds of thousands of years without any energy input, which is absolutely true. You find these fossilized creatures, it's very resilient. And stored in a capsule a few centimeters in diameter, it can hold up to 500 terabytes of digital data. By 2025, the total volume of digital data generated by humans is expected to reach 175 zettabytes. And I looked it up for comparison. 1 zettabyte, I think, is a billion terabytes. Yes, 1 zettabyte is 1 billion terabytes. So that's a reference point. And since the current storage mediums are relatively bulky, fragile, and energy intensive, meaning just to keep the hard drives, at least when they're in operation mode, running, they take energy. And when they're not, they are subject to, I would imagine, more entropy, more decay than something more organic would be. Something that seems to be amazingly resilient. What do they say? Preserved for hundreds of thousands of years versus, material that breaks down. Rather than these fragile, energy-intensive, and bulky storage systems, DNA may provide a viable alternative, able to contain all existing data within a space of a shoebox. So if I'm understanding that correctly, if we utilize DNA, all the data that we have on Earth, I suppose, is what they're saying, would fit inside of a shoebox, which is pretty amazing. Facilitating DNA storage will be this initiative, PEPR, Molecular Archive goal, a priority research program provided last May by the French National Center for Scientific Research. So yeah, just very, very cool recent development in the use of DNA as a storage device. This sort of thing, like I said, blows my mind. You can imagine, you can follow a clear logical path as to seeing that, okay, obviously this would be something that we would figure out how to do eventually, but the logistics of how it's happening, the actual mechanics, is just kind of mind-blowing to me. Not just that they do it, oh, that's crazy, these scientists, they can do anything, but just the ingenuity and the ways of hacking these biological machines that we are, and finding the replicatable features and tethering them together in little chains to do things like storing massive amounts of data onto DNA and then retrieving it or filtering it and getting your results through fluorescent signals. It's all kind of crazy to me. But maybe that's me. Maybe, you don't care about how much, storage you can, store in DNA. And maybe this is mind blowing to you. Let me know your thoughts in the comments, and I will talk to you soon. Cheers. Bye.